right, so it's great to see a wonderful packed room. Very pleased to present to you the Michael Johnson Seminar speaker for today. So I had the pleasure of meeting Mike almost 10 years ago during the Multicultural Scholars interview. After the interview, we always had to fill in our feedback. And I distinctly remember there's only been one time that I've ever used all caps, please have this student come. They're a wonderful human and then be a great scientist. So Mike grew up here in St. Louis, and he knew early on that he wanted to be a scientist. He enrolled in Maryville in 2017, pursuing biochemistry and data science. During his time at Maryville, he completed 2 internships, one in elemental enzymes as a biochemist, and one at CTC Genomics as a gene editing engineer. He pursued part-time research with Dr. Fox Budich for 2 years, working on atomic spectroscopy, and 3.5 years in my lab working to understand telomere extension drug mechanisms of action. In 2021, after graduating from the BS from Maryville, Mike went to the University of North Carolina, Chapel Hill, in the lab of Sean Ahmed, where he studied telomere binding proteins in C. elegans via microscopy and genomics. In 2022, Mike started his doctoral work at the University of Colorado in Denver, where he currently resides in the lab of Gregory Clay. Mike is currently working on pioneering new methods in high throughput, high content, image-based drug screening. It's with immense pride that I introduced our Michael Johnson Seminar speaker for January, Mike Lippincott.
Thank you for the kind introduction. Nice to be back at Hill. Just so I have a sense who is here in terms of what majors and their real paths. Not everyone all at once.
And I'm here for the University of folks.
Just one per table so I can get a broad sense of the topology of the group. I'll start calling on people.
Biology, pre-PA program.
Physical therapy.
Biology.
Third.
Science.
Did I miss the table? Yeah, I did it with back one.
Biochemistry.
Yes, for all science. Great. You're in the right room. Yeah, so go ahead and get started. So just so we have some definitions out there on the table. Some of these words can be used interchangeably and they're not always. So disease. Says when we lose cellular homeostasis. What is a pharmaceutical or a drug in this sense? It's a compound manufactured from use as an additional drug. The cure is different from pharmaceutical. This is a substance or treatment that actually cures a disease. Completely restoring cellular homeostasis. Which the treatment is an attempt to get a closer. So the mission that we take in the way what I'm doing with my dissertation work is to reduce human suffering caused by disease. My goal is to restore cellular homeostasis to treat or cure disease. So what this looks like from an abstract point of view is we can view some sort of map where cells can exist. We can have a control state. These are healthy cells. We can have disease states. We can have intermediate states. And there's paths and trajectories all along that a cell can exist and bounce between the states. So a treatment would be bumping a diseased state cell to more intermediate state, not quite all the way back to most. And cure bumps you all the way back to that state. It's very different, and it's a continued discretizing or placing cells into bins, but instead there's a whole distribution. So we've all heard of the central dogma, but this is how I view it. It's very so not linear. It's a fully connected graph where there's multiple nodes. We have 23,000 roughly genes in DNA that we code for over 30,000 RNA, maybe more. Hundreds of thousands of proteins. We can adjust those proteins. And then there's a bunch of other crap We're talking lipids, carbohydrates, metabolites. There's all of this stuff going on in the cell. It's really not as linear as those textbooks might say. DNA, RNA, protein have one potential path through this structure. So the way I want everyone to restructure how we think about cells is a fully connected complex graph where every single molecule is interacting with all of the other molecules. We're just all big into each other and mix out. So if you add all of these up, there's an immense amount of molecules in the cell. And this is ultimately what dictates how cell behaves, how a cell looks, and how a cell is or isn't distinct. And so the way that we can study cells classically is to rip it down and tear it apart and look at all of the individual pieces, right? That's what Legos are. But you couldn't necessarily tell me what this looked like before I dropped the Lego set. So we can sort of think of cells as assembled Legos and all the components individually as Lego pieces, and they amount to the greater whole. And so understanding how two pieces fit together can be very important, but understanding how those two pieces fit together within the context of a whole cell, that can only be done in certain contexts. So how do we do that? So this whole notion that size and shape equals function of a protein applies to many things outside of biology and biology. So we have this DNA, RNA, protein, other crap is fully connected sort of things. So that's a phenotype. And that phenotype, the cell, we can call it. This is a concept posed by a scientist in Waddington back in 1907. We can use a morphology of the landscape, the topology of all of these cells, with all of these underpinnings of the other crap, the DNA, the RNA, the protein. And as those change, so did everything else within this. So how do we study how all of those individual pieces back to be larger cell or the organism? Well, we do that via process. We've all seen what a microscope looks like, but what they don't tell you, and like you won't always learn right off the bat, so this isn't really what a microscope looks like. This is more of what a microscope looks like. These are $500 million to $1 million instruments to view inside of a cell to really understand the phenotype and the morphology of how a cell is changing when we factor these two different elements. And it gets even crazier. Some microscopes you can't buy, you have to build them yourself. So these are all individual systems that a single lab might possess that they've built through their own methods. And what it allows you to do is collect these really quite insane images.
Power does it
Power.
Yeah, I mean, like, many folks like
We don't think of things as power or magnification, but rather raw resolution. And so you can get down to the refraction of lights, depending on the wavelength in which you're measuring, depends on how well it is to yourself. So you're looking at 400 nanometers of wavelength. It's like this. So with super res, you can get sub-100 in resolution on the pixel basis in this image. So what I'm interested in is how these molecular organelle networks change the morphology. This stuff in the cell, how does that change the morphology, and can use that morphology to better screen illness, which is their drug screens. And we can. In every great drug screen, you have to have an assay. And the assay that we use is cell painting. So we can stain nuclei, actin, Golgi, plasma membrane, RNA, nucleoli, endoplasmic reticulum, mitochondria. So we can stain all these different components of the cells. This is pretty pared down for cell painting. As of 2 years ago, there's in-plex experiments where you can stain every organelle ever discovered in the cell, so up to like orders of 48 plex, so you have 48 semester going on. Well, what this allows us to do, I'll show you a more blown up a couple cell, which you can really see the individual mitochondria networks, the ER networks, where the Golgi lie outside of the cell. And this really starts to look like the diagram of cell we see in textbooks, which is, I always thought those were just made up. So we use cell painting to screen for drugs and for treatments. And you're saying, well, how do we go from this picture to understanding how a drug interacts or works? So if it works. And I would say that's a very good question. Full marks. So to do that, we turn to computer outputs. So now that we have images, all these images are arrays. This is linear algebra. These are arrays of numbers. These are instead of ones and zeros, these are values. It's a measurement of how many photons has hit a camera detector. It's just how your camera works. And so the darker something is on this picture, the less light it has received, the brighter it is. So now we're not thinking of cells as cells at all. We're thinking about them as numbers. And when you reduce everything to numbers, you can just do math. That makes things easy. So for the computer to be able to do anything meaningful, we have to tell the computer where to look in the image. So we perform something called segmentations. We can go in and understand where the nuclei are within the cell. And that's informative to tell us where the individual cells are. Now we sort of have this outline, like a feed almost of where to look in the image on what a cell is and what a cell isn't. We can take measurements of those. We can also look at where the cytoplasm is by simply subtracting the nuclear space from the cellular space. You know, everything that exists around. So when I say featureization from here on out, really what I mean is we're taking measurements. It could be as simple as how wide and long is the cell, what's the diameter, things like that, to more complex measurements where we can get to multiple orders of complexity. Say, what's the distribution of a given signal in this object? What is the size of a given object within that cell? How punctated is it? How granular is it? Is the texture smooth or is the texture very rough? Do we have these Zernike features, these borders that emerge, geometric patterns? These are all things that we can measure that tell us something about the cell and the system that we're studying. So the way that we do this is we have this full open source software stack that we've developed in the lab that we can from raw images. We go through, we perform multiple steps of QC, elimination correction, segmentation, feature extraction, or measuring of the cells. We can then go through this software stack essentially, and at the end of this whole process, what we get out is this tabular format. So a table where every single row is a single cell for hundreds of thousands of images. And then we have each of those features for measurements, measuring thousands of these features that describe what each of those cells are. So we essentially quantified or digitalized the cell into numbers. Now we can perform complex data science up to design multiple experiments to test how do cells under one drug treatment differ from a control. Okay, that's a lot of backgrounds, getting everyone up to speed for what I'm doing, why I'm doing it, how I do it. So if there's any questions, we can answer that before we go into more of what we've been able to do so far with this.
When you said you give the computer data to look up
look out for the segmentation pieces. What is this type of data that you're asking us to look out for?
Yeah, so it's all microscopy images. Two more. So essentially, this is what I feed the computer, and this is what the computer spits out. And I use that very vaguely, saying, get it to the computer and the computer give it back to me. I'm telling the computer what to do here by writing algorithms, software, coding, right? So it's multiple lines of code to do this. Computers are very literal, they do exactly what we tell them to do. Anyone that has written any code knows that if you write an error in your code and you run it again, you get the same error. It's never the computer's fault, it's always going through a fault.
How susceptible is this error, like in identification or like misidentifying certain cellular components?
Very.
That's gotta make your work hard. More difficult than other ones.
Yeah, there's headaches at times getting this to work. There's a lot of optimization for those things. Run this on one cell type, if you change cell types. These are neuroblastoma cells isolated from pediatric patients. But now if you take, say, a blood cell, with a bunch of blood cells in there, very different. Yeah, it's really.
So for the, I'm assuming that this image right here is like it's the output segmentation, right?
Yeah, overlaid onto the.
Overlaid on the
Yeah.
cell, on the nuclear, right? So. Is this the output of like one of those softwares that we were showing?
No, this is the output of like just segmentation of things.
So for the segmentation algorithm, and it's okay if that's somebody else's work, but for the segmentation algorithm, is it like computer vision, image processing, or there's some AI involved in doing this segmentation?
Used to be no, it used to be all flow based. And you would compute gradients and process the signals, but now mostly everything is just the same, the segment and model framework, which is self-supervised distillation technique where it works. It's pretty much gold standard best in class, and everyone's using it. The issue with that versus this doesn't hallucinate, but the Dino framework, because it's a transformer, it hallucinates and there'll be random blobs
Yes.
to in missegmentation, so you have to run a QC step.
It's very susceptible to this, yeah, that's why I asked.
Yeah, so it's like pros and cons. I would say it's like, okay, do I drive the NASCAR car to work today or do I take a Prius?
Yeah, yeah.
I just had a good question based on the microscope fact that you pointed out early on, stating that oftentimes when it comes to analyzing your cell segmentation, things like that, there's a lot of times where that has to actually be built. Maybe was there ever a time during your analysis where you actually worked with others or you had to create a version of the microscope so that you would be able to, you know, analyze further the things you're looking for?
Yeah, I built one about 4 years ago. It's like a $250,000 system. That was a lot of fun, but it does help. Like when you build it, you know all the specs inside out. And when you're doing this analysis, sometimes you have to know the exact distances between your samples and the lens and like how far the light actually travels inside. Those sorts of things. So when you build it, you know all those things. And it's but if you don't build it, you can just ask whoever the microscope needs. They should know it. I'm going to go with that. All right, is that better? Better. There's a question over here. No.
Are we there yet at the high-dimensional rate?
No, so this technically is what I would call 3D data, because there's a spectral component to it, but everyone refers to it as 2D, which is X, Y.
So means that when we go to high dimensional data, we actually look into the cell.
Yeah, we're technically doing that here, we're looking into like a cross section of the cell, it's like, you rip open a house of a dollhouse and like a King Kong. Like, and, yeah, it's essentially what we're doing. Anything else? Okay, great, wonderful questions. Now we'll move into more of the, how we apply this to higher dimensional. So. Looking at using this specifically for understanding apoptosis is really key. Some of my prior work is working on a different form of cell death called pyroptosis, and this is an inflammatory form of cell death. Apoptosis is not, but everyone here has heard of apoptosis, so that's what I'm talking about. So a little bit of a controversial opinion here, but all disease is essentially, does a cell die when it's not supposed to, or does it not die when it is supposed to? So regulating cell death is part of cell homeostasis, so it's really important to a study. And so we'll be looking at how do we study apoptosis and how do we do that through this time. We can use one of these microscopes that I showed earlier. We can apply a 10-point dosage curve with gyrosporine, which is a protein kinase inhibitor, wreaks havoc on the cell. The cells will die, and I'll show you what that looks like in a little bit. But for the drug screens, you can imagine we can take a 96-well plate like this, and for multiple wells, we can take multiple images in each of those. When you do that through disease machines, go different steps through the test. In this case, we max project them because we don't hold their thick sample, and so we end up with 2D time lapse that we process it. So we use these live cell dyes called chromolive. They're minimal toxic, and it allows us to go through and view how each of the compartments, specifically here and gold here, are changing through time. So we can take a picture every 30 minutes for 6 hours. Then we can run through our typical pipeline. Into our nation correction, which is just QC mark. We can perform cell and nuclei segmentation. We then go through and track each of these cells. And actually assign a single node or point to every cell and every frame, and then connect them through a fully connected graph and then solve that. And now you can go through and you can follow a cell as it's moving even through like every 30 minutes. So this is really powerful. You then run our featureization. We do this on each of those andron features. We also feed this into a dynatherm that I was talking about earlier. This is essentially a black box model where we end up with per channel 384 features that we have no clue what they need, but they are measuring something about the pixels within that given cell. If you ever hear someone say that AI is a black box, this is a great example of that. Then we do our image-based profiling, and through our software stack that gives us our cell by quality feature matrices. So here's just some representative images of how these cells are changing over time at a couple of different doses of cephalosporin. And this is what you have to do. Published papers have to be static, but this is a talk, so it's really nice is we can just play videos of these. I think it's a lot better. So just pick one square and watch it for the whole duration of 6 hours, and you can see how even by eye these cells are changing quite drastically. You can see in the upper left is our control condition. There's a lot of changes, but they're very different changes from, say, a highest dose of cephalosporin and even some of those middle doses of cephalosporin. So this is an experiment of multiple controls. What's really great is we can quantify everything. We can take that matrix that's thousands of features long, form a dimensionality reduction technique called RUMA. And this essentially is taking all those features, they're not shown, collapsing them down into 2 axes, and we can then visualize how these are changing through time. So every time step, we can see how these points are shifted from control to a very high dose of status quo. We're effectively capturing how these cells are changing through time. So, how many people have seen this before? Okay, a couple of words, excellent. So the story behind this is in World War II, all of these generals wanted to essentially beef up armor for some of the planes. They were coming back with all these bullet holes everywhere, and I don't know if it was like a 1-star, like 5-star general, but like someone essentially said that we should add armor to where all the bullet holes were. And the statistician, if you were involved, you can actually see all of his calculations, they're public online, it's really cool. He said, well, no, we shouldn't add armor to where the bullets are, because we observed every single one of those planes making it back to base. We should be adding armor to all of the places where the bullet holes aren't, because they were the planes that didn't make it back. So those planes all crashed, they got shot there. So makes sense, right? This is a concept in statistics called survivorship bias, where you're really only measuring cells that are alive, but also dead, right? So this is really crucial for time-lapse studies. A lot of the ways people are doing drug screens by performing cell painting, waiting say 6, 24, and 48 hours, running, taking images, and say, yeah, there's like some fewer cells, and the cells look different. You're really only captured by the cells that are like after they're dead, not the process of undergoing apoptosis. And so it's again survivorship bias. Depending on cell type, this can be even more dangerous. Let's say you're studying other form of cell that I studied, pyroptosis. These are all immune active cells, so they're macrophages. What do macrophages do to dead cells? They go through and they eat them up. So you're not going to have any evidence after 48 hours that say cells died, because the ones that didn't die, you're now studying the wrong thing, and yeah, it's just a headache. So this is really the sort of thought process behind why we profile for drugs in a time lapse. This is really sort of on the edge of this research field and pushing it forward with these frameworks have been also. Okay. Any questions on the time-lapse stuff, and then we'll jump into 3D and volumetric data.
So as you were saying about like the cells viewing them normally, if like someone's getting tested for some disease or something, are you saying that this could lead to a lot of misdiagnoses, or like your new like thing could improve diagnosis of the accuracy of what's actually happening if you're looking at actual stuff?
Yeah, maybe not diagnoses, but definitely in terms of like a drug discovery framework. Say you test drug X, and after 48 hours, you see that there's no difference between like the control condition. That drug doesn't make it through the drug discovery pipeline. That is a 20-year-long process, and it gets moved out pretty early on in that process too, versus if now you're running some sort of mathematical model, we could say, oh, there was a change, and there was a huge change at hour 6 of that 48-hour time point, and then we'll see this sort of recovery. Now you're recovering that biological signal that you wouldn't have otherwise.
So instead of just looking at beginning to end, it'll show.
Yeah, it's like running around a track, and let's say it takes you 2 minutes to run a quarter mile. I take a picture at 0 seconds, and I take a picture at 2 minutes, and I said, she went, oh. Which is not true.
For a time lapse, what were we, when the data was from like on the X and Y axis, what were those, and is it showing like one feature or like a representation of multiple actually?
Yeah, that's exactly right. It's a representation of multiple segments. So UMAP's one of those things where like I don't think anyone actually knows how it works exactly, except the people that wrote this paper. It's a unifold, unifold. No, sorry, uniform manifold approximation projection is what UMass is for. And so essentially what they're doing is taking this like high-dimensional data space, putting it into a trash compactor and then saying, here's the representation of this. This these columns are like these like matrices are like thousands of cells, and that's what a data point is. But then the measurements that they're taking are like thousands. So like how do you plot 1,000 axes? Another classic way to do this is through PCA. But there's UMath is the like gold standard for visualization. You're only doing analysis on this after like you win PCA, although some people do, and I think they're wrong. But that. Okay. Moving forward in time into 3D. So the next part of my work we'll talk about is the 3D frameworks that I've been setting up for getting out of a 2D context and into a volumetric. So if I throw a pathway like this up here, it's pseudo comprehensible, and unless like you're an expert on the RAS pathway, all of these pathways are like alphabet soup, right? So essentially, when we look at RAS, it has this GTP and GDP, and the GTP state is active, so that propagates its signal forward and goes through this whole cascade, right? We have one molecule and protein bumps into another, usually by phosphorylation events, and what that results in is cell substance. We have this other protein called neurofibrillin. Neurofibrillin has a core function of going through and removing phosphate group from GDP turning into GDP. It's very similar to how ATP works. It's just instead of adenosine and blood. So what happens if we get rid of neurofibrillin or we lose just one copy? We end up, this is what we call haploinsufficiency. We lose one copy, we don't have enough, and that results in the phenotype. So what happens is if you're lacking one copy and you're haploinsufficient for N1, you're going to have neurofibromatosis type 1 or F1. And this results in all of these growths all over the body, on the skin and the neural sheets. These tumors can become very large, like baseball and softball size. They're can grow in large abundance, and they're incredibly hard for surgeons to resect or operate on, especially the ones that surround or touch our feet. They also grow in the optical neuron pathways, so most patients with NF1 will have one or both as this is something a lot of times the patients are born with, and there's no cure for it, and the quality of life is not very good. So there's two pharmaceuticals on the market right now, so selumetinib and mirdametinib. They both target MEK, so it's downstream of RAS. So regardless if you have enough neurofibrillin or not, you can inhibit MEK, and then you can thus inhibit this overabundance of cell growth. And this sort of helps with some of these inoperable tumors, but this doesn't work in every patient. Just like any other drug doesn't work with every patient. And it's incredibly costly. Selumetinib, the compound, is $250,000 for a lab. So $250,000 is a lot for it to make it not work. And selumetinib can only be used in adults, not children. For NF1 children with these tumors also. So this is how we get around this. We need new treatment options, we need more drugs, and we need to screen them, and we need to be screening them like 10 years ago. So that's where we come in and take all of these plates from patient-derived organoids. So we collaborated with multiple surgeons that have these tumors out, the ones that they can. We get that material, we can cryopreserve them. And we can grow in each one of these wells hundreds of microtumors essentially, where we can screen all of these drugs. Very similar to the time lapse where each well have hundreds of organoids in, and then you can take multiple patients within that well after applying some sort of drug preparation. So we can scale this up for multiple patients or as many drugs as we can use for as many as we can use. We can perform cell painting on these, and we can perform cell segmentation and image-based profiling to get these morphological features. So the process haven't changed, but the technical parts have. So when we image these, we image them volumetrically. So the way that we do that is by optically slicing. So the microscope focuses at different depths. It's like when our eyes do it automatically, but if you focus on this desk, go with that desk, go with that desk, this stuff is out of focus. I focus on this desk, that's out of focus. It's all of the microscopes doing. We're changing our focal lengths. Does anyone do photography in here?
You might depth of field. The depth of field, right?
Yeah.
We
You can have it open or closed. So that's essentially all we're doing, is changing the focal. Just taking all of these images, we call them slices, and we can stitch them all together and generate these volumes.
like you check the aperture.
Yes, yeah, essentially. Yeah, you're just going through, it's not aperture, like a camera, because the lenses on microscopes don't have axes. But essentially it's, yeah, we're actually just physically moving.
Well, have a steady note.
Yeah, it's like if you have your focus here and then you keep that focus and you just move that thing. That's what we're doing on the microscope. So again, we can perform cell paintings that nuclei, actin, Golgi, plasma membrane, ER, and cytochronic membrane. So once again, we have to tell the computer where to look. This right here, getting the computer to see and segment individual nuclei and organoids and cells is about 6 months of work. To write the code for this, it's probably 10 to 15,000 lines of code to be able to do this. AI doesn't quite do this yet. This biological data is quite unique to other 3D models. If you look at Facebook or Meta, their models are concerned with how I do things in 3D from a single image, that it's everything in focus. This data is not the 2D image looking at 3D space, but rather it is volume. This is an unsolved problem that we're still working towards, but a lot of the work has been really diving into how do we actually segment these objects in 3D so that computers can actually tag them. We can also do this for each cell, right? So we can propagate a signal from each of the nuclei. Where does the signal start? Is the signal stopped? And that's how we segment each of these cells. And this process is not only has it taken a long time to develop, but it's very computational and intense. This is about a 30 micron deep organoid, and so picture that that's about 30 times the amount of data as a single image. So now we have to have the compute power to do all that. So we run all of these analyses, all of this segmentation, the featurization, all that is critical. It's just it was essentially just thousands of computers strung together and
When you go to the high dimension, actually we have like a exponential of data.
yeah, we have a linear increase in data and exponential increase in data processing. So we can take the combinations of each of these masks and each of the channels, and we can compute very similar features to 2D. We get area size shape, which is looking at surface area and volume, and we can look at how the centers of mass of the signal shift, we can get co-localizing the signals, we can look at granularity and intensity, we can compute neighbor analysis, so we can understand where within an organ a cell is. So say it's a cell more internal or external, what's the distance of that cell from the center of the organ? Is it more dissociated or is it more compact? So has the treatment actually inhibited the growth of this organ, or has it completely killed dissociated these cells, and how they're not so gravity quite tight on? So we've done this on a very small selection of drugs so far as proof of concept. The next step in this project is to really scale this up to a larger drug screen, but what we have so far early on in this work is essentially a way to go through and understand on a per patient level, how do these patients respond to these different drugs. And so what I'm showing you here is mean and average precision, and you can think of this as a metric of you compare a morphology profile compared to the control. How different are they? So if they're really similar, we're gonna have an NP score of 0, so that means that the drug effectively isn't working, there's no difference. And if we're at 1, that drug cannot be more different. It is the most different that they could possibly be. And so essentially, and then the y-axis is just the significance of that, how confident are we that that is true. And so essentially, what this gives us is on a per patient level, we can screen drugs that we believe will actually perform in the clinic. So this hasn't been validated yet, but we've had 8 patients worth of data, and we're getting another 3 or 4 coming in. They're processing now actually. And so we can see if you look at N014 in the upper left here, we have a way bigger difference in, say, rapamycin versus we look over at N021, that sort of blue is actually quite more shifted to the left. So you can just compare each of these from patient to patient, and you can see, okay, each of these patients are responding quite differently to each of these drugs. So what we think we have here is potentially a new framework for precision medicine for these patients to say, okay, Solumet, is it likely to work or not? Because it's $250,000 to compounds, we kind of want to have a little bit of confidence on it's going to work. And from, say, a sample from the patient from these organoids and imaging, it only takes 1 week. And then for each patient to process everything through, it takes another week. So for some of these tumors that aren't quite as aggressive as others, a 2-week delay in decision for how to treat a patient might not be that big of a deal. It's probably be certainly be more cost effective, but if it works better, then I think that'd be good. Also, something I didn't mention is these drugs, Solumet, is incredibly messy. 90% of the patients lose their vision while undergoing treatment. Solumet, and about 10% lose their vision entirely, and 80% get their vision back. So while you lose your vision while you're on this, you really don't know if you're gonna get it back or. So it's, we preferably not want to use that drug, but.
Is 25% precision really that good of a standard for drugs? I don't know. No experience.
Yeah, so. It's better than 0. Right.
Fair enough.
And so it's all on a relative scale, so. This is only a small handful of drugs, and a lot of them have very similar targets. They're all in the fast pathway, upstream or downstream, so they're all performing pretty similar. So we're not really getting any information out of this. It's really more proof of concept for can we do this. But yeah, I think it's not a good score. Where you set that threshold or cutoff, then that's a good question. We don't know because we don't have a positive control, right? We don't have healthy versions of these cells that we're aspiring to be, like we do other projects, so like, it's really hard, but I think what we can do is say what goes with each of them. It's maybe not one of the needs. But yeah, it's a good question. Yeah, and so with that, everything we've been doing is open source, publicly available, it's all on GitHub, our data sets, you can download it, terabytes worth of images, you can download them. And we perform team science, so everything that we do in the lab development is collaboration with other labs all around the world. So this has been a lot of fun. Yeah, I mean, we love the people from multiple continents, it's absolutely fast. We might start something up with the folks down in Australia. It's just difficult to like time all the time zones. And here's are all the collaborators that we collaborate with. These are just some of the teams, not the people they collaborate with too. So it's a much larger network. But rarely anything that you do in science is just you or just you and your advisor. I think the things worth doing require more than one or two people. And yeah, it's a lot of fun because every person you interact with, you get to learn from things. That's kind of the whole point of science, is to learn something. At least in my opinion. So yeah, with that, that's everything that I have for you today. I'll take any questions.
Do you think that this type of data analysis can be used for interesting analysis and as one, like other cell duplicating diseases that we don't necessarily understand? And
Yeah,
that.
yeah, and that's what we're hoping, is to get more disease specific models in 3D. So one of my friends studies ovarian development, and she did all of this work. We're hoping we can apply this, and she studies specifically fertility questions, and then how do these cells turn over, when do they turn over, and what happens when they senesce. This would be a really good way to answer some of those questions, so we can target it towards where we want. The hardest part after you have all this developed is now you have to grow the cells, and you have to grow the cells in a fish that actually fish likes how they grow. So if you don't do that, everything that you collect is pretty much meaningless.
So how do you replicate those conditions? How do you-
Yeah, so that's a lot of trial in there. Large factors are things like extracellular matrix. So depending on where a cell might be in the body, what you culture your cells on will be really important. So the things like blood cells and macrophages, they have a very diverse environment that they interact with. In bloodstream, which can be speaking through cells that have very high extracellular matrix values that have very stiff environments. Muscle cells, those a lot of times can be very stiff, but too stiff, and so like there's a lot of work going on there versus like epithelial cells tend to be locked in, but a little more close to UC. So a lot of it is studying ex vivo systems, like take a sample from a living organism, like a mouse or a histology sample, and you can replicate certain conditions. And a lot of that is comparative and a little touchy feely. It's not like a one size fits all. It's kind of where science and art intermeet.
So, I remember you had a slide, that was talking about featurization, and it had like, low and high neighbor features, right? So, when you talk about temporal data, right, like, are you calculating, like, are you doing that featurization at each of those, right? That's why it's so high dimensional. For each cell, you have all of that featurization, but you also have that for every time instance, right?
Yeah, and the benefit to doing it that way is that now you can plug it into any model. Like you can run it as a discrete, you run time as a discrete variable, as a continuous variable, you can just an ordinary differential equation, we use a stochastic differential equation, we still have that data. Did it through versus if you do all that at the time of featureization, you're pretty locked in. And depending on the experiment, let's say we're comparing pyroptosis versus apoptosis. Pyroptosis occurs very fast, it's part of the immune system. Apoptosis is much slow. If you're capturing one in 15 minutes and one in 30 minutes, and you're capturing the features on different time scales, and the time scales are intrinsic to the features, now you have it's a nightmare. I won't touch it. So it's like you want these discrete time points that you could interpolate and make continuous or these differential equations later on. And that's where the fun is, but like you gotta actually collect that correctly.
Oh, my question was just, since this is only being used for like looking at drug accuracy, like how it works in patients, do you think there's a future for this to be used in patients that aren't diagnosed, like the leading up to that?
Yes, I think that's up to the FDA. Well, ultimately, like, there's obviously a validation mark that needs to occur for all of this. What we don't know, like, what we think we know, but what hasn't been outright proven or a lack of disproof is that whatever score that we generate for a patient to respond well to a drug, that they actually do, that hasn't been validated. So we'll need some sort of to actually assess that, which I don't think will be that hard to do in terms of, because these are, keep in mind, all the drugs that I showed are already FDA. We know that they're safe in patients and that they can be effective. So this is, I think, a very low risk.
Like, as well as this imaging, is this available for like research of diseases, like if you're looking at cancer and stuff like that, this would be a good.
Yeah, I mean, that's primarily used for research rather than diagnostics. But yeah, you can do it. You can use this to study anything you want to study. And one of the benefits of cell painting that I didn't talk about in imaging is once you have your microscope, you're good. Cell painting costs pennies on the dollar compared to if you're going to run a sequencing experiment or, God forbid, you run a proteomics experiment. Those are very expensive. Other hands on there. There wasn't AI hallucinating. Implement AI models.
I have a question. I know you mentioned that there are many of you all working together as each team, those across the country, things like that, other parts of the country. When it comes to a project like this that takes so much time and tedious amount of work in different areas just to be able to conduct all this research, how does that, like, how do you all work together in order to accomplish as much as you have presented, it's presented here today on what you all have studied? How is that broken down? Like, are there roles that, some of you have to, let's say, for instance, you create the program, the coding that you need to do this, or those who do the math, working of it all. How does that work when it comes to that networking teamwork that you all have to do?
Yeah, I really like that question. So my work starts when I get images. So a lot of the work that I did before I started my doctoral work was actually with imaging. So I have a lot of experience with that. Some of it here at Maryville, we have both licenses here still. Yeah. Okay. Yeah, so like you can start this stuff, maybe not tomorrow, but the day after. So like, but yeah, so we have like a wet lab team and a dry lab team. I want dry lab team, so I'll do all of the coding for this, all of the data visualization, the data analysis. That all starts when I get the images. It's a lot of work, but so is the other part of the work, which is generating the data, especially if multiple patients, and there's people in that group, so their whole job is to coordinate the clinic to the lab. And yeah, it's a 60-hour-a-week job. You have to know when the patients are scheduled, which patients have consented to have their samples placed in lab. You have to get release forms. But like, there's so many, everyone has their job to play, and it's all. You know, but the good news is you don't have to do it all, and should, because. Well, you might get it with that, but it's like finding what you wanna do and how other people will implement that. Yeah, I think just like being a part of it is fun, so like you can find, carve out your niche where like you can contribute.